Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be explaining you fatty acid synthesis, particularly de novo synthesis of fatty acids. Let's get into details. When a person is in fed condition and when a person is relaxing and resting, especially after carbohydrate meal, and when insulin levels are high and glucose levels are high, at that time a fatty acid synthesis will be going on mainly in the liver, in the lactating mammary glands, and to a certain extent in the adipose tissue, brain and kidneys. Now there are several steps that can be involved in the fatty acid synthesis. So formation of fatty acids from a metabolic intermediates, that is a de novo biosynthesis of fatty acids, can be divided into three stages. One is a transport of acetyl-CoA carbons from the matrix of mitochondria into the cytosol. The number two is conversion of acetyl-CoA carbons into malonyl-CoA carbons. And the third is reactions that are going on in the fatty acid synthase complex. So now let's get into biosynthesis of fatty acids in each one of these three stages. Now the first stage is a transport of acetyl-CoA carbons from the mitochondrial matrix into the cytosol. Now note that whenever a person is in well-fed condition and when insulin levels are high and when a person is in restful condition or resting, at that time especially the glucose, which is at higher level in the blood, enters into the liver, and in the liver glucose is converted into glucose 6-phosphate, and that will be done by 𝗒𝐿-CoA enzyme. And now 𝗒𝐿-CoA enzyme, it is induced by insulin, because under fed condition, if the person is in fed condition for a long period of time, so 𝗒𝐿-CoA is induced by insulin. Now glucose, which is converted into glucose 6-phosphate, and glucose 6-phosphate will be converted into fructose 6-phosphate, and fructose 6-phosphate further goes into glycolysis and converted into fructose 1,6-bisphosphate. And this fructose 1,6-bisphosphate, it is also indirectly kept active by insulin. And then fructose 1,6-bisphosphate is broken down into glyceroldehyde 3-phosphate and dihydroxyacetone phosphate. And then further glyceroldehyde 3-phosphate goes all the way down into phosphoenol pyruvate, and the phosphoenol pyruvate is converted to pyruvate by pyruvate kinase enzyme. Now the pyruvate kinase enzyme is also kept active by insulin. Now once we get pyruvate gets down into the mitochondrial matrix, and in the mitochondrial matrix this pyruvate is converted into acetyl-CoA molecule, and the conversion of pyruvate into acetyl-CoA, it will be done by pyruvate dehydrogenase enzyme, and that pyruvate dehydrogenase enzyme is also induced by insulin. Now once we get acetyl-CoA, and acetyl-CoA is going to condense with oxaloacetate in the matrix of mitochondria to form citrate. Now note that when the person is in well-fed condition and resting, so there will be plenty of NADH plus molecules in the matrix of mitochondria, because electron transport chain is not running at higher level. That means higher levels of NADH plus will have a negative effect on certain enzymes in the T-cell cycle, especially three enzymes in the T-cell cycle, which are negatively modulated by higher levels of NADH plus in the matrix of mitochondria. Those are isocitrate dehydrogenase enzyme, alpha-ketoglutarate dehydrogenase enzyme, and malate dehydrogenase enzyme. All these enzymes will have a negative effect by higher levels of NADH plus, and that's why what will happen is isocitrate builds up, that will be reverted back into citrate formation, and citrate will build up in the matrix of mitochondria now. And this citrate will be taken out of mitochondria into the cytoplasm by tricarboxylate transporter. Now that is what is shown here. So the tricarboxylate transporter moving a citrate into the cytoplasm, and in the cytoplasm citrate will break into acetyl-CoA and oxaloacetate. That will be done by an enzyme called citrate lyase. Now what happens to the oxaloacetate and what happens to the acetyl-CoA? Oxaloacetate, it will be converted into malate, and that malate, it will be converted back into pyruvate done by maleic enzyme. And during this process, there will be formation of NADPH plus H plus molecule. And this NADPH H plus will be used for reductive biosynthesis, and that is what is shown here with the maleic enzyme. So, oxaloacetate to malate and malate going into maleic enzyme reaction, giving NADPH H plus. Here is the NADPH H plus that is shown, converting malate into pyruvate, and pyruvate coming down into the matrix of mitochondria. That's what happens to the oxaloacetate there. So what happens to the acetyl-CoA? This acetyl-CoA here, which came out of mitochondrial matrix in the form of citrate, because citrate is the one that carries carbons of acetyl-CoA from the matrix of mitochondria into the cytoplasm. So in the cytoplasm, acetyl-CoA will get into fatty acid synthesis reaction. So the reaction that is shown here is a acetyl-CoA carboxylase enzyme catalyzed reaction, which is making malonyl-CoA. So acetyl-CoA converted into malonyl-CoA. That means 2 carbon acetyl-CoA converted into 3 carbon malonyl-CoA. Now this 3 carbon malonyl-CoA will get into fatty acid synthase enzyme catalyzed reaction. Multiple reactions will go on here, ultimately making 16 carbon palmitic acid. And that's how carbons of acetyl-CoA from the mitochondrial matrix will be transported into the cytoplasm in the form of citrate, and that is done by tricarboxylic acid transporter. And citrate breaks into acetyl-CoA and oxaloacetate, providing acetyl-CoA for reductive biosynthesis, that is fatty acid biosynthesis. Some of the citrate in the cytoplasm can be converted into isocitrate, and isocitrate is converted into alpha-ketoglutarate in the cytoplasm done by isocitrate dehydrogenase, that is cytoplasmic isocitrate dehydrogenase there. And in this process, there will be generation of NADPH plus H plus. That's another source of NADPH H plus for the reductive biosynthesis, which is shown in the figure here. And also note that the glucose 6-phosphate that is elevated whenever glucose comes in and converted into glucose 6-phosphate, and this glucose 6-phosphate, not only it is going into glycolysis, some of the glucose 6-phosphate is diverted into pentose phosphate pathway. And in the pentose phosphate pathway, especially in the oxidative phase of pentose phosphate pathway, so there will be generation of NADPH plus H plus molecule. So there are 2 enzymes in the oxidative phase of pentose phosphate pathway. One is glucose 6-phosphate dehydrogenase, other is 6-phosphoglucanolactone dehydrogenase. Both of them will release NADPH H plus, and these NADPH H plus will go into reductive biosynthesis process. So this is how a transport of acetyl-CoA carbons from the matrix of mitochondria into the cytoplasm will go on, especially in the form of citrate. And in the cytoplasm, the citrate will break open into acetyl-CoA and oxaloacetate, and that will promote a fatty acid synthesis because the reactions that I have explained so far will provide all the necessary requirements for fatty acid synthesis. What are the necessary things for fatty acid synthesis? The substrate for fatty acid synthesis is acetyl-CoA, which brings 2 carbons, and the coenzyme or the reductive molecule for fatty acid synthesis is NADPH plus H plus. Alright, let me give you the sources of NADPH H plus which are needed for fatty acid synthesis, or for that matter the reductive biosynthesis that is going on in the cytoplasm. That is, fatty acid synthesis is a reductive biosynthesis, cholesterol biosynthesis, bile acid synthesis. So all these type of reductive biosynthesis needs NADPH+. So what are the enzymes that will provide NADPH+ in the cytoplasm? So they are, I'm going to write the list here. Number 1 is pentose phosphate pathway enzyme glucose 6-phosphate dehydrogenase. I'll write it as G6PD. Number 2 is 6-phosphoglucanolactone dehydrogenase. That is the second enzyme in the oxidative phase of pentose phosphate pathway. Number 3 is maleic enzyme. Maleic enzyme is, it is converting malate into pyruvate. That's the maleic enzyme which I have just now explained. And the number 4 is cytoplasmic isocitrate dehydrogenase. So these are the 4 enzymes that will provide a source of NADPH plus H plus. Let me highlight that for your convenience here. So NADPH H plus is coming from pentose phosphate pathway, and that's number 1, pentose phosphate pathway, source number 1. So there are 2 enzymes in pentose phosphate pathway, especially the oxidative phase. That is glucose 6-phosphate dehydrogenase, second is 6-phosphoglucanolactone dehydrogenase. Now the second source is maleic enzyme. Maleic enzyme is second source of NADPH plus, number 2 there. Okay, let me write it number 2 here. Now, what the maleic enzyme does? Maleic enzyme, it is going to convert malate into pyruvate and doing the decarboxylation and releasing NADPH plus. Now number 3. So maleic enzyme is number 3. Number 4. Number 4 is isocitrate dehydrogenase in the cytoplasm. So it is going to convert isocitrate into alpha ketoglutarate in the cytoplasm, making NADPH plus H plus. So that's number 4. So like this, there are 4 sources of NADPH plus in the cytoplasm which will facilitate NADPH requirement for reductive biosynthesis like fatty acid synthesis. Okay. Now, let me explain you what is the role of insulin in promoting fatty acid synthesis. So I will highlight those things as I move on. Just now how I did for NADPH source. So, conversion of glucose into glucose 6-phosphate, which is done by glucokinase enzyme. Glucokinase, and this particular enzyme is induced by insulin. So let's write that here on the side. So insulin, it is going to promote fatty acid synthesis at various levels. So starting from conversion of glucose into glucose 6-phosphate, making sure more and more glucose gets into glycolysis to provide pyruvate converting into acetyl-CoA, because acetyl-CoA is the carbon that are coming out into the cytoplasm in the form of citrate to go into fatty acid synthesis. So insulin has to promote all the enzymes to go there. Okay. So glucokinase enzyme, that's number 1. Insulin is going to increase glucokinase enzyme activity, and then the second stage is PFK1 enzyme. That is the master controller of glycolysis, PFK1. It is indirectly promoted by insulin by keeping higher levels of fructose 2,6 bisphosphate with the PFK2 enzyme. So I'll write it as PFK1 and PFK2 enzyme. PFK1 and PFK2 promoted by insulin. Now, as we move down, so we have a pyruvate kinase enzyme here. That pyruvate kinase also kept active by insulin. So let's add that into the list. Number 3, pyruvate kinase. Pyruvate kinase enzyme, it is kept active by insulin, so thereby promoting glycolysis and making sure carbons of glucose are taken towards fatty acid synthesis. Once pyruvate is made in the cytoplasm, so this pyruvate coming down into the matrix of mitochondria and now converted into acetyl-CoA, and that is done by pyruvate dehydrogenase enzyme complex, And this pyruvate dehydrogenase enzyme complex, it is kept active by insulin. So I'll add that as number 4. So it is PDC, pyruvate dehydrogenase complex. Now the acetyl-CoA, which is having those 2 carbons coming from glucose, so it is going to condense with oxaloacetate to make citrate. Now the citrate is the one that is moving out of mitochondria into the cytoplasm. So that's how citrate, the acetyl-CoA carbons from the mitochondria transported out into the cytoplasm, done by tricarboxylate transporter. Now the citrate breaks into acetyl-CoA in the cytoplasm, that's what is shown here, and oxaloacetate carbons are separated, done by citrate lyase enzyme. And now insulin is going to keep this citrate lyase active. Citrate lyase enzyme is kept active by insulin. Now oxaloacetate, which is there in the cytoplasm, will be converted to malate, and the malate will be converted to pyruvate, done by malic enzyme. And now the malic enzyme is also kept active by insulin. So number 6, malic enzyme. So what is the role of malic enzyme? Malic enzyme is going to convert malate into pyruvate and give NADPHH+. So insulin is going to make sure the NADPHH+ requirement for fatty acid synthesis is met. Okay. Now on the other side, what happened to the acetyl-CoA here, which came into the cytoplasm? So acetyl-CoA is getting into malonyl-CoA formation, and that is done by acetyl-CoA carboxylase enzyme, and that enzyme is also kept active by insulin. So I'll add acetyl-CoA carboxylase here. And then malonyl-CoA, once it is made, so malonyl-CoA will go into fatty acid synthase enzyme complex, and that entire fatty acid synthase enzyme complex, it is kept active by or induced by insulin. So FA synthase, fatty acid synthase complex, is induced by insulin. And then all the other means the pentose phosphate pathway enzymes, 2 of them, that is glucose 6-phosphate dehydrogenase and 6-phosphoglucanolactone dehydrogenase. Those 2 enzymes are also induced by insulin. So I'll write 9 and 10 here. 9 and 10. Those are pentose phosphate pathway enzymes. So like this, insulin will make sure that all the necessary requirements for fatty acid synthesis is made available, so that in the cytoplasm fatty acid synthesis will go on. So this is what is the first stage here in the transport of acetyl-CoA carbons from the mitochondrial matrix into the cytoplasm, and then how these acetyl-CoA carbons in the cytoplasm are going towards a fatty acid synthesis. Now let's move on to see how a conversion of acetyl-CoA carbons to malonyl-CoA carbons happens. The conversion of acetyl-CoA to malonyl-CoA. So acetyl-CoA is converted into malonyl-CoA by acetyl-CoA carboxylase enzyme, and acetyl-CoA carboxylase, it catalyzes a committed step in the fatty acid synthesis. So as it is shown here in the figure down here, acetyl-CoA, which is a 2-carbon molecule, will be converted into malonyl-CoA, which is a 3-carbon molecule. Now take a look at the color coding. So acetyl-CoA, black colored carbons there, and one of the carbon in a malonyl-CoA is colored blue, and that blue color is coming from carbon dioxide, indicating that acetyl-CoA carboxylase enzyme is incorporating a carbon dioxide into the molecule as carboxyl group there. So the malonyl-CoA carboxylic group, the 3rd carbon in malonyl-CoA, it is actually the carbon coming from carbon dioxide. So remember that color there. So since acetyl-CoA carboxylase catalyzes the committed step in the fatty acid synthesis, so this is also one of the most regulated step in a fatty acid synthesis. Meaning the citrate, whenever citrate is present in the cytoplasm, insulin is going to send that acetyl-CoA, which is converted to citrate moving out of mitochondria into the cytoplasm by tricarboxylate transporter, and the citrate in the cytoplasm, which is available there. So it is going to act as a positive modulator on acetyl-CoA carboxylase. Although citrate breaks into acetyl-CoA and oxaloacetate to give acetyl-CoA carbons in the cytoplasm, some of the citrate will act as a positive allosteric modulator on acetyl-CoA carboxylase in the cytoplasm. That's what is shown here. And the end product of fatty acid synthesis is a long chain fatty acyl-CoA, and that long chain fatty acyl-CoA will act as a negative modulator on acetyl-CoA carboxylase. So how this happens? So citrate, what it does, it causes a polymerization of acetyl-CoA carboxylase and make it as an active polymer. Whereas long chain fatty acyl-CoA, it will inhibit that polymerization process and just maintain the inactive dimeric form of acetyl-CoA carboxylase. That's how it happens. So anyway, acetyl-CoA carboxylase, what it does, it is increasing the concentration of malonyl-CoA in the cytoplasm. Now malonyl-CoA is going into fatty acid synthesis. That's one thing. So the second thing is this increased malonyl-CoA levels will have a negative effect on CPT1 in the outer mitochondrial membrane. Note that the CPT1 is carnitine palmitoyl transferase 1 in the outer mitochondrial membrane is necessary for the transport of long chain fatty acids into the mitochondrial matrix for beta oxidation process. So presence of higher levels of malonyl-CoA in the cytoplasm will have a negative effect on CPT1, thereby it is going to decrease beta oxidation of fatty acid. Beta oxidation of fatty acid is decreased because we are looking at fatty acid synthesis. So when fatty acid synthesis is going on, fatty acid oxidation has to be decreased. Remember that. Okay. So this is about the conversion of acetyl-CoA into malonyl-CoA done by acetyl-CoA carboxylase. Now let me explain you regulation of fatty acid synthesis. So the acetyl-CoA carboxylase is the most regulated enzyme in a fatty acid synthesis pathway. So how this acetyl-CoA carboxylase is regulated? So the acetyl-CoA carboxylase regulated by allosteric mechanism and also by hormonal mechanism. So what is the allosteric mechanism of regulating acetyl-CoA carboxylase here? So hormonally, the citrate is acting as a positive modulator on acetyl-CoA carboxylase and thereby maintaining the acetyl-CoA carboxylase in a polymeric state, whereas the end product malonyl-CoA, it is a negative modulator of acetyl-CoA carboxylase, thereby acetyl-CoA carboxylase levels will decrease. So that's about allosteric regulation. Now what is the hormonal regulation? Hormonal regulation is insulin. Whenever person is in well-fed condition, insulin is going to activate protein phosphatase enzyme. And this protein phosphatase enzyme is going to remove phosphate from the surface of acetyl-CoA carboxylase and maintain the enzyme in a dephosphorylated state. And the dephosphorylated state of enzyme is an active state of enzyme. And also insulin is going to induce acetyl-CoA carboxylase enzyme, thereby increase the concentration of acetyl-CoA carboxylase, and thereby it is going to increase a conversion of acetyl-CoA into malonyl-CoA. Now what about the antagonistic hormones? Antagonistic hormones here is glucagon and epinephrine. Both glucagon and epinephrine through protein kinase A enzyme, they are going to phosphorylate acetyl-CoA carboxylase enzyme, and the phosphorylated form of acetyl-CoA carboxylase, it is an inactive form of enzyme. That's how glucagon and epinephrine will maintain enzyme in inactive form. Similarly, during exercise, so AMP which is rising, it's going to activate AMP activated protein kinase, and that is going to phosphorylate acetyl-CoA carboxylase and keep it inactive. So this is how acetyl-CoA carboxylase, which is the major regulatory enzyme in fatty acid synthesis, is regulated allosterically and also hormonally. Alright, now let's move on to see a fatty acid synthase complex. As you see here, a fatty acid synthase complex, it's a complex enzyme having 14 enzyme subunits and also 2 protein subunits. So on either side, you are going to see 7 enzymes and 1 protein on the left side and also on the right side. So fatty acid synthase complex is divided in the center as a functional division. On the left side, you are going to see 7 enzymes and 1 protein, which is ACP. On the right side subunit, you are going to see same 7 enzymes and 1 protein, that is ACP. Now, on either side, both the left side, we have one set of enzymes to make a fatty acid. On the right side, we have another set of enzymes to make a fatty acid. So that means in the fatty acid synthesis, there will be 2 fatty acids that are synthesized simultaneously. So when fatty acid synthase is working, there will be 2 fatty acids synthesis going on, one on the left side and one on the right side. Okay, so we'll have to have that understanding as we move on. And also note that all set of enzymes are available in one place, and that too in pairs. So, and the regulation of all these enzymes becomes easy because they are all present in one protein complex, and the regulation of the gene for them is much easier. So the coordination of the reaction becomes much easier, and that's why the efficiency of fatty acid synthesis will become much more better. So that's why fatty acid synthesis is a highly efficient process, and this entire fatty acid synthase complex is induced by insulin. Remember this. Now let's move on to see how exactly fatty acid is synthesized on either side of this huge fatty acid synthase complex. Now the figure here is showing you how a fatty acid synthesis is going on using a fatty acid synthase complex. So what I will do here is I am going to divide this fatty acid synthase into 2 halves, one on the left side and other is on the right side. Now what is explained or what is shown here on the right side, same thing will be going on the left side. That means you should understand, so the things that are shown on the right side, all the reactions that are shown on the right side, so similar reactions are going on the left side. That means 2 fatty acids are synthesized simultaneously. Now the very first thing that goes on, very first reaction that goes on here with the fatty acid synthase enzyme complex is acetyl-CoA getting into the reaction. And note that the acetyl-CoA carbons are mentioned in red color. Okay, you got to pay attention to that color coding. I'm going to highlight that with green, right? So let me just take that green out. So you see the red color there. Okay, these are red color acetyl-CoA, carbons in acetyl-CoA, original acetyl-CoA molecule, red in color. So the acetyl-CoA carbons are attached to ACP. So now once that happens, those acetyl-CoA carbons which are attached at ACP, they will be moved to the cysteine position. Now the ACP position is vacant because the carbons of acetyl-CoA, the 2 carbons of acetyl-CoA which are moved to the cysteine position, ACP position is free, and to that malonyl-CoA carbons are attached. Now the malonyl-CoA coming in there, and out of 3 carbons in a malonyl-CoA, 2 carbons are actually from acetyl-CoA done by acetyl-CoA carboxylase, and the 3rd carbon is from carbon dioxide, and that is colored blue. Malonyl-CoA molecule is attached to the ACP. In the cysteine position, we have acetyl-CoA carbons which are red in color. So now what happens? The acetyl-CoA carbon is having a nucleophilic attack on the malonyl-CoA carbon, especially the 3rd carbon of malonyl-CoA. Now the 3rd carbon of malonyl-CoA, which is a carboxyl carbon there, so it will be released as carbon dioxide. So the carbon dioxide released here, it is blue in color, meaning it is the same carbon dioxide that is added by acetyl-CoA carboxylase enzyme. to make malonyl-CoA. So the carbon dioxide which was added by acetyl-CoA carboxylase to make malonyl-CoA, same carboxyl group here in the malonyl-CoA will be released as carbon dioxide. So meaning carbon dioxide can never become part of a fatty acid because it is released here in this reaction. And this reaction is referred as condensation and decarboxylation reaction. So it is condensation and decarboxylation. Condensation and decarboxylation reaction where acetyl-CoA carbons having a nucleophilic attack on the malonyl-CoA carbons. Now we have a 4-carbon molecule attached to ACP position. Now if you look at it carefully, so we have a methyl group and then we have a keto group there, and we don't want a keto group in that position because we are going towards a fatty acid synthesis, and then we have CH2 and the carboxyl group. Now the keto group which is present in the 3rd carbon here, because this will be number 1 carbon, number 2, number 3 and number 4. Now the 3rd carbon keto group need to be reduced, and that will be done by using NADPH plus H plus, and that is a reduction reaction there. So reduction. So we have done condensation, decarboxylation. Now there is a reduction. So reduction of keto group into hydroxyl group here. Now hydroxyl group is also not required here because we are going towards the fatty acid synthesis, where in the fatty acid we have a hydrocarbon chain, methyl end and carboxyl end. There is no hydroxyl group there. So hydroxyl group has to be removed, and that is done by dehydration. So dehydration. This is the 3rd reaction in the sequence. So first one is condensation, decarboxylation. Second is reduction reaction, and the 3rd is dehydration. So after dehydration, there is an introduction of double bond here between 2nd and 3rd carbon. But our intention here is to make a saturated fatty acid. So there should not be any double bond. So this double bond need to be reduced, and that is using one more NADPH plus. So the reduction is going on now. So that means double bond between 2nd and carbon is reduced. Now you have this organic molecule which can be released as a fatty acid. So it has got number 1, number 2, number 3 and number 4 carbon. So this is a 4 carbon fatty acid can be released now, which will be a saturated fatty acid. So like this, acetyl-CoA carbons, 2 carbon from acetyl-CoA, original acetyl-CoA carbon, red in color, will be attached, which will be transferred to ACP position, and then later transferred to cysteine position, and to that AC position malonyl-CoA carbons are brought in. Malonyl-CoA is a 3 carbon molecule. Now the nucleophilic attack happens between the acetyl-CoA carbons which is attacking a malonyl-CoA carbon, and that makes release of carbon dioxide, which is the same carbon dioxide that was added by acetyl-CoA carboxylase enzyme. So that means carbon dioxide can never become part of a fatty acid. So that is referred as condensation decarboxylation reaction, number 1 in the sequence of reaction here. And then keto group is reduced into hydroxyl group, done by reduction process using NADPH plus molecule. So that is the 2nd in the series of reaction. And now we have a hydroxyl group which has to be removed. That is done by dehydration reaction, number 3 here. And then we have a double bond which need to be reduced into single bond there. So that is done by the 2nd reduction reaction using NADPH plus. So after this we have a 4 carbon molecule which can be released as a fatty acid. Note that in that 4 carbon molecule, last 2 carbons are red in color, 3rd and 4th carbon red in color, right? And the 1st and 2nd carbons are black in color. So it means 2nd, 3rd and 4th carbons are coming from acetyl-CoA, and the 1st and 2nd carbons are coming from malonyl-CoA, which is there in the number 3 reaction. There is malonyl-CoA. So now this 4 carbon fatty acid which can be released now. So generally what happens in our cells, there will be continuation of reaction. That means this 4 carbon molecule will be shifted to cysteine position. Now the ACP position becomes vacant. To that we are bringing one more malonyl-CoA which contains 3 carbon. Now there will be a nucleophilic attack of 4 carbon molecule to a 3 carbon malonyl-CoA carbons. So there will be condensation and decarboxylation. Same process will be repeated now. Reduction where NADPH is used, dehydration and then reduction. So at the end of condensation, reduction, dehydration and reduction, now we get a 6 carbon molecule. So you number the carbons here. So number 1 carbon, number 2, number 3, number 4, number 5, number 6. Look at number 5 and 6, they are red in color. That means they came from acetyl-CoA. Rest 4 carbons from 1 to 4, they are from malonyl-CoA. So one thing that you need to note here is the acetyl-CoA carbons, they are pushed towards the other side, towards the methyl end. The meaning that malonyl-CoA carbons are added every time towards the carboxyl carbon. Always remember that. In a fatty acid synthesis, malonyl-CoA carbons are added to the carboxyl side of a fatty acid, and the acetyl-CoA carbons, they are all moved towards the methyl end. So the red color carbons that you are seeing here, so previously in a 4 carbon molecule, they were at number 3 and number 4 position. Now in a 6 carbon molecule that is made now in the second spiral of reaction. So now it is in the present, it is at 5th and 6th. So 3rd and 4th now moved to 5th and 6th. So that means the position moved by 2 carbons, because 2 carbons are added towards the carboxyl side and we are numbering carbons from the carboxyl side. Okay, like this a 6 carbon fatty acid is made now, and it just continues for few more spiral of reaction. So that's a second one. Now we are going to repeat these spirals. So far we have number 1 and number 2 spirals. So 2 spirals happened. Now the number 3 spiral, number 4, number 5, number 6 and number 7. 7 spirals. In these 7 spirals, every time in each spiral there will be addition of 2 carbons. That means 6 carbon fatty acid at the end of 3rd spiral become 8 carbon, and then the 10 carbon, then 12 carbon, then 14 carbon and then 16 carbon. So at the end of 7th spiral there will be 16 carbon palmitate which is made, and finally there will be release of this palmitate molecule from fatty acid synthase complex. So like this, a saturated 16 carbon fatty acid is synthesized using a fatty acid synthase complex. So some of the things that you need to be careful here in understanding fatty acid synthase complex and reactions and how a palmitic acid is synthesized, although it looks very complicated, but if you look at it carefully, so it is not as complicated as it looks like. Let me just explain you one more time in a really a simple way so that you can get the take-home message. The take-home message here is to understand how these carbons are added in a series of reactions. Okay, let's begin only by highlighting certain things. So look at this acetyl-CoA carbons which are red in color. So track that red colors there. Acetyl-CoA carbons are in red color. So bringing 2 carbons into fatty acid synthesis. That's the only time original acetyl-CoA enters into fatty acid synthesis. Remember that point. Okay. And then we have a malonyl-CoA here. This malonyl-CoA, it has a 3 carbon, and out of these 3 carbon, one of the carbon will be released as carbon dioxide after that condensation and decarboxylation reaction. I'll write it as C and D, condensation and decarboxylation. There will be release of carbon dioxide. So meaning carbon dioxide is not being part of a fatty acid being synthesized. Okay. And now after this condensation and decarboxylation, So keto group is converted to hydroxyl group is reduced into double bond is converted into a saturated single bond. Okay. Now, so that means there will be condensation, reduction. In the reduction, there will be use of one NADPH, then the dehydration, and then one more reduction. Another NADPH is used. So basically, when a 2 carbon condensing with another 2 carbon, making a 4 carbon molecule, and that 4 carbon is all the way reduced to have a 4 carbon fatty acid which is attached to the fatty acid synthase now. So it is going to use 2 NADPH to make a 4 carbon. 2 NADPH plus molecules are used so far. After that, so this 4 carbon molecule which is moving into cysteine position, so to the ACP position, another malonyl-CoA comes in. So now you see malonyl-CoA enters into the fatty acid synthesis all the time. Acetyl-CoA comes in only once, malonyl-CoA comes later. So in the first spiral, this is the number 1 spiral, one malonyl-CoA coming in, and note that this malonyl-CoA actually it is synthesized by acetyl-CoA carboxylase. I will show it here only. So acetyl-CoA, which is a 2 carbon molecule, is converted into malonyl-CoA after carbon dioxide is incorporated there. So I will color that carbon dioxide here, CO2, done by acetyl-CoA carboxylase. Okay, so that's how malonyl-CoA comes in. So in this process of conversion of acetyl-CoA to malonyl-CoA, ATP is used. One ATP is used here. Okay, so I'll just put that circle there, red circle, ATP. One ATP is used to make one malonyl-CoA. So every malonyl-CoA that is consumed, there is a consumption of one ATP. Remember that. It can be asked in the exam. So acetyl-CoA to malonyl-CoA done by acetyl-CoA carboxylase. ATP is consumed, carbon dioxide is incorporated. That malonyl-CoA getting into fatty acid synthase complex. So the carbon dioxide that was incorporated by acetyl-CoA carboxylase will be released in the condensation decarboxylation reaction. So the carbon dioxide cannot become part of a fatty acid. Okay. So in that spiral, one malonyl-CoA is used. Now in the second spiral, so in the fatty acid, like 4 carbon fatty acid is getting into the second spiral. So here is the second spiral. In the second spiral, there will be another malonyl-CoA used. That means one more ATP is used, because every malonyl-CoA means an ATP being consumed there, because to make malonyl-CoA, one ATP is used, right? So that malonyl-CoA in the second spiral will give 2 more carbons, and these carbons are added towards the carboxyl side, and the methyl side is just pulled on the other side. It is just going, the number of position changes. So that means red color carbons are just pushed aside. Okay. So like that, every spiral, one malonyl-CoA comes in, and each spiral there will be use of 2 NADPH molecules. How many spirals? Multiplied by that many NADPH molecules. Let's see how many spirals to make a palmitic acid. So now so far we have 2 spirals. So as we move on, so right now we have 6 carbon molecule which is attached here. Now the third spiral, fourth spiral, fifth, sixth and seventh spiral. In each spiral there will be addition of 2 carbons, right? So 16 carbon means 16 divided by 2. So that means you need 7 spirals to make a palmitic acid. Okay. So in each spiral I told you there is a consumption of 1 malonyl-CoA. That means if it is a 7th spiral, 7 malonyl-CoAs are used. That means 7 ATPs are used. So if you know the number of spiral, then you know how many malonyl-CoAs are used to make that fatty acid. How to know the number of spiral? So you know the total number of carbons that are there in the fatty acid being synthesized. Divide that total number by 2. So we are going to get 8 there, right? So that's the number. So 8 out of 8, so that means 8 acetyl-CoA molecules. So 16 divided by 2 is 8, and 8 minus 1 will give you the number of spiral. Or other way, what you can do, you use this formula, 16 divided by 2 minus 1. to know the number of spirals. So you are going to get 7 there. And this is the 7 number that will give you. So the number of spirals in a fatty acid synthesis means number of malonyl-CoAs that are getting into fatty acid synthesis. So that is the number of ATPs being consumed. So 1 malonyl-CoA means 1 ATP being used. 7 malonyl-CoAs means 7 ATPs are used. And also note that 7 spirals in a fatty acid synthesis means in each spiral of fatty acid synthesis 2 NADPH plus molecules are used, because there are 2 reduction reactions. You know, condensation, decarboxylation, reduction, dehydration, reduction. So in those 2 reduction reactions, so 1 NADPH is used, so 2 NADPH used per spiral. So 7 spiral means 7 multiplied by 2, 14 NADPH plus used. So that's another thing that you need to remember. And also you need to locate the carbon source in a final product, that is a 16 carbon palmitic acid. So in a 16 carbon palmitic acid, so where is the acetyl-CoA carbons which are red in color? So I told you, every time this red color carbons, acetyl-CoA carbons, are pushed on the other side. So in 4 carbon fatty acid, they were at 3rd and 4th position. In a 6 carbon fatty acid, they were at 5th and 6th position, right? So like that, they are pushed towards the end. That means in a 16 carbon fatty acid, the red color carbons will be at 15 and 16 carbons. That's the position of a red color acetyl-CoA carbons. Rest of all carbons are actually came into the fatty acid synthesis as malonyl-CoA carbons. Remember that. So number 1 to number 14 carbons in a palmitic acid is malonyl-CoA carbon. Number 15 and 16 in a palmitic acid is a acetyl-CoA carbon. So that's another thing you got to remember there. So these are some of the things that you got to remember in a fatty acid synthesis. It's a quite complex pathway, looks like that, but if you break it down and take the message out of it, so that makes it easier. So now let me give you the take-home message for fatty acid synthesis chapter. Make sure you remember the things here. These are the high yield points. Now the acetyl-CoA carboxylase catalyzes a rate limiting reaction in a fatty acid synthesis, and also it is the most regulated enzyme in fatty acid synthesis. And the regulatory molecules for acetyl-CoA carboxylase is citrate is a negative modulator, long chain fatty acyl-CoA is a positive modulator. Hormonally, insulin is going to keep acetyl-CoA carboxylase active by maintaining it in dephosphorylated state, whereas glucagon epinephrine will keep it in a phosphorylated state and it's an inactive state of enzyme. And also insulin is going to induce acetyl-CoA carboxylase enzyme. Now the fatty acid synthase complex, it's a multi-subunit enzyme with 2 functional domains, and having all the enzyme subunits in one place makes it highly efficient and regulation becomes easier. Insulin is going to induce this entire enzyme complex. And the malonyl-CoA, it is used during fatty acid synthesis. It is synthesized from, it is coming from the acetyl-CoA and carbon dioxide. That is done by acetyl-CoA carboxylase enzyme using 1 ATP there. And the carbon dioxide present in the malonyl-CoA is not incorporated into fatty acids but are released at condensation and decarboxylation reaction. That means carbon dioxide can never become part of a fatty acid. And also in a fatty acid synthesis process, there are 4 reactions which are repeated every time, and those are condensation and decarboxylation, reduction, dehydration and reduction. Now all carbons of fatty acids are made from malonyl-CoA carbons from 1st carbon to 14 carbon, except the ultimate and penultimate carbons of fatty acids which are originally coming from acetyl-CoA. So let me change the number here. So if it is a 16 carbon fatty acid that we are looking at, so number 1 to number 14 carbon, 1 to 14 carbon are coming from malonyl-CoA carbons, and this 15 and 16 carbon are coming from original acetyl-CoA carbon. So, in a fatty acid, last two carbons, that is ultimate and penultimate carbon, last two carbons in a fatty acid towards the methyl end. See the methyl carbon and the carbon besides that, last two carbons are coming from acetyl-CoA. Rest all carbons starting from the carboxyl carbon all the way to last but one carbon, and after that, like in a 16 carbon, first to 14 carbon, they are coming from malonyl-CoA carbons. Remember that. And each cycle of fatty acid chain synthesis, whenever malonyl-CoA is incorporated, so it needs 2 NADPH plus H plus molecules. That means in a palmitic acid synthesis, there will be 14 NADPH and 14 H plus molecules are used. That means if you know the number of spirals that are needed to synthesize a fatty acid, so that many malonyl-CoAs will be used and that many ATPs are used, and 2 times of that NADPH plus will be used. That means for a 16 carbon fatty acid, so the number of spiral will be 16 divided by 2 minus 1, that will be 7. So that means 7 malonyl-CoAs are used. So that means 7 malonyl-CoAs are used means 7 ATPs are used and 14 NADPH plus 14 H plus molecules are used. So double that spiral, you will get the number of NADPH plus. So remember that. And these 7 malonyl-CoAs will contribute to carbons number 1 to 14, and the acetyl-CoA will contribute to carbon number 15 and 16 in a 16 carbon palmitic acid. And the acetyl-CoA carboxylase is the most regulated enzyme in a fatty acid synthesis. It is allosterically modulated and also it is hormonally modulated. So these are all the points that are related with fatty acid synthesis. So that's about fatty acid synthesis, especially synthesis of a saturated fatty acid, which is referred as a de novo synthesis of a fatty acid. Thank you.